third chapter in the cardiovascular system is the blood vessels and the circulatory system. Okay, how it circulates around. We'll talk about the different vessels, talk about pressure, the control mechanism for blood pressure and circulation and that kind of thing. Okay, so we've talked about arteries, capillaries, and veins before, as far as arteries carrying blood away from the heart, veins carrying blood back to the heart, and the capillaries where exchange occurs. And that's the most important one when you think about the vessels is the capillaries, because that's where we're exchanging things. Otherwise, it's just in a tube being carried somewhere. So the capillaries, smallest blood vessels, and where we have exchange between the blood and the interstitial fluid. You could say extracellular fluid. Okay, so either one of those terms work for this blood that's, or this fluid that's outside of the actual capillary that's leaked out of the bloodstream. Okay, but our other two, arterioles and venules, these are the smallest branches of arteries for arterioles, and the venules are going to be just on the other side of the capillary as far as the smallest of the veins that are going to start the return process. As far as the blood vessels go, the largest of our blood vessels are going to attach directly to the heart, be it an artery or a vein. Our two largest arteries, that pulmonary trunk and that aorta, and then the veins when we think about superior vena cava, inferior vena cava, that kind of thing. Okay, smallest ones are going to be capillaries. We can't see those. Those are microscopic. Okay, we'll look at some in a slide that I'll show you a little later on. Okay, so these vessels, they have similar structures. When we look at these, and on our diagram, let me get my pen out here, we've got an artery over here, we've got the vein over here, and then when we're looking at them side by side, and that's the way that they run side by side, or brachial artery next to the brachial vein, right? Axillary artery next to the axillary vein, okay? Things like, it's always gonna run in pairs like this. So if we're comparing them, notice that it's different here. The lumen of the vein is quite different from the lumen of the artery. Okay, we're going to see lots of differences here, and that's one of the questions that I've asked back when we weren't doing just all multiple choice tests. But I would say, give me 5 differences between arteries and veins. Okay, so let's take a look at this. Okay, we've got 3 different layers that both arteries and veins have. Okay, there is the tunica intima. That is the one that is closest to the blood. It's intimate with the blood, we say. So it's going to consist of this epithelial layer here and a little connective tissue behind it, the tunica media, and then the tunica externa. Okay, same thing over here. Tunica intima, tunica media, tunica externa. Okay, so when we are identifying these, and you'll be doing this under the microscope from a microscope slide. You don't have a microscope at home, but I'll show you some pictures of these, and you're going to tell me whether it's an artery or a vein on the lab test. Okay, so the way that we can tell the difference, there are some big differences here. The vein is usually, excuse me, up here, the vein is usually very oblong, flattened out structure because it's under very low pressure. The artery, much more round. The artery is typically the smaller vessel, vein is larger, and when we look at the walls of the vessels, the artery is much thicker. See all this area here makes up the walls here, and then the thin vein. Again, this is due to the pressure differences that we see between arteries and veins. The artery has a much thicker tunica media because it has to have more control due to the pressure. Okay, there are one of the ways that I look at them a lot of times under the microscope is if we have this rippled endothelial layer. Okay, because of this smooth muscle here, there's always some kind of squeeze going on our blood vessels unless we are in some type of shock. Anaphylactic shock will open up all those blood vessels. So because it's under some kind of squeeze, there's this rippling effect that happens on the inside where the endothelium is. Notice out here we don't have as much control, so it's going to be a very smooth endothelial layer. Okay, and you can kind of catch some of that rippling right in this area here on our picture. Okay, Collagen and elastic fibers we're going to find in the artery. We've got collagen and elastic fibers out here, and even a little bit of smooth muscle out here in the vein, but a much smaller level here. Okay, two other things that we can identify very easily under the microscope. If we were in lab, I'd have you be looking at these. That is the presence of this structure here, the internal elastic membrane, as well as the external elastic membrane. It's going to kind of sandwich that tunica media layer. It's a squiggly line that we see here and that we see here on the outside, not a squiggly on the outside. Okay, but it gives a lot more elasticity. And again, that pressure is going to mean we need a lot of elasticity here. It's not about elastic here because we're in such low pressure. Okay. All right, let's take a look. Arteries and veins. Tunica intima, we said interlayer here, and we are looking at an artery here. So tunica intima, it's going to have an endothelial lining and a connective tissue, okay, backer. We think about it like a membrane almost, okay. Then second layer, tunica media, all smooth muscle, okay, concentric sheets of smooth muscle. We've got an internal and an external elastic membrane that can give it a little stretch as well. And then tunica externa out here, collagen elastic fibers. We see a little bit of smooth muscle out here in the veins. So that tunica externa is what attaches the vessels to their location, okay? It kind of cements them into that area, okay? It says that they run side by side. We can look at the difference here, see the much thicker wall and the round shape of the artery, okay? You can see the collapsed look of our vein, okay? And let's see. Much greater volume here too, right? Low pressure, greater volume, high pressure, less volume. Okay, so like we said, the veins lining contracts, which means we don't get the rippling effect here. The artery lining creates the folds, those ripples that we looked at on the inside here. A lot more elasticity here. And the last big thing, veins have valves. They're under such low pressure, we have to put valves here to prevent backflow of blood. And I'll talk to you about those. Okay. So if we're talking about pressure, we're typically talking about high pressure versus low pressure. High pressure is going to be on the arterial side. Okay, that's where we're going to have the greatest amount of contractibility. We're able to change diameters a lot more in the arteries than we are in the veins. And this is controlled by our sympathetic division of the autonomic nervous system. Okay, we talked about this in A&P1 when we got to the brain and how the sympathetic division of the autonomic nervous system can help us contract if we're in fight or flight mode. Okay, and it's going to allow us to move blood wherever we need to move it in the body. Okay, if you constrict it, if you do vasoconstriction, okay, that's going to be a less volume, so we're not having as much blood flow go to that area. If we do vasodilation, we're opening up that area, enlarging the lumen, and providing more blood to that location. And we see this all the time. We see as things happen, we're able to move blood to an area, and that part of our body will become a little bit redder in an area due to this increase in blood flow. So when we compare them, this is what they look like, okay? Up close to the heart, they're going to be the biggest, and these are called the elastic artery, and if you're a vein, you're just called a large vein. And then as we move away from the heart, we get to a second type. That's the muscular artery. And what is the venous side for a muscular artery would be a medium-sized vein. And then we've talked about arterioles and venules. Okay, and then we're going to talk about our different types of capillaries. And we've got several different types of capillaries to talk about here. Okay, so elastic arteries. So here's our blood. Elastic arteries are going to be those that are the closest to the heart. Okay, so when we think about arteries characteristics, we've got elastic arteries, we've got muscular arteries, and we've got arterioles. And they're going to decrease in size as we move from the heart down to the capillaries. Okay, first one, elastic arteries. These are the ones that are the closest to the heart. These are also called the conducting arteries. So things like the aorta and the pulmonary trunk. Okay, and if you look at this, notice that there's not a tremendous amount of smooth muscle here compared to there is down here. Okay, it's about the same. That's because our elastic arteries are not really ones that we want to control a lot. Okay, we want them to have a full volume of blood that we can distribute throughout the body. So their job is mainly to stretch, and that's why they're called elastic arteries. Okay, this elasticity, as the blood moves into that area of the artery, it's going to cause that artery to expand, and the elastic fibers, like the elasticity in your underwear, pushes back against it. Like that's what holds it onto our bodies. Okay, so when we pushes back, it kind of helps even out this pulse force. Okay, so this elasticity is important for us. From the elastic arteries, we move to the muscular arteries. Okay, muscular arteries, these are also called distributional arteries. Okay, still have our three layers: tunica media, tunica intima on the inside, tunica externa on the outside. Okay, the muscular arteries are, like I said, distributional arteries, meaning these are the ones that we've got tremendous control over. Lots of smooth muscle here in the middle for us to dial the blood down if we want to restrict blood flow to an area, relax and do vasodilation if we want to move more blood to a particular area. So that's the role of these. Then the arterioles. Okay, when we get down to the arterioles, these are small. We've lost the tunica externa. Okay, the tunica media is still there, but it's very thin. Okay, we still have our endothelial layer. Our whole tunica intima is still intact. Okay, and these are much smaller and much more branched. So as we go, we have very few elastic arteries. We have more muscular arteries as they branch away, and then even more arterioles as they continue to branch and get smaller and smaller and smaller. So that diameter is important here. The diameter is important. And remember, we're not exchanging things if we're in the elastic artery. We're not exchanging things if we are in a muscular artery. We're not exchanging things if we're in an arteriole. There's too much stuff around the vessel to prevent oxygen to be able to make its way through, for nutrients, for waste to be able to come in. Okay, so that sympathetic nervous system, okay, is going to determine where we need oxygen. If we get low blood oxygen levels in an area, if those tissues aren't getting enough oxygen, that causes sympathetic division of our autonomic nervous system to send more blood to the area that needs it the most. So just by tapping your foot, if you start tapping your foot, you need more blood flow to that foot. Okay, and if we constrict those arteries, we're opposing blood flow. We're making it harder. We're dialing down the diameter of the lumen of that vessel. It's going to take a lot more pressure, so the blood's going to want to go the other direction. Okay, so we made it through the arteries all the way down, arterioles. Now we've got the capillaries to talk about. And the capillaries, like I said earlier, these are the most important of the vessels. I can't stress that enough. These are the most important because this is where exchange is going to occur. The whole reason to put something in the bloodstream is so that we can take it out somewhere else. We can put oxygen in it in the lungs so that we can take it out of the tissues, right? We load it up in the digestive system the blood with nutrients so that we can take it out when we get to the different parts of the body. Okay, we load the amount of waste that's on the outside has to be loaded up and moved in the bloodstream so that we can filter it with our kidneys. Okay, endocrine glands, they have to put the hormones into the bloodstream so they can go wherever they need to in the body and cause things to happen. So we got to have these capillaries. This is where things are leaving the bloodstream. This is where things are entering the bloodstream. It's got to be in a capillary. It can't be in any of the other vessels. Okay. All right. So these capillaries, microscopic networks, okay, permeate all the active tissue. Okay. So if we say it's vascular tissue, that means it's got capillaries. Remember, epithelial tissue was avascular, so we got no capillaries in the epithelial tissue. Okay. We have to get it from underneath. The function of the capillary allow for two-way exchange. Two-way exchange, meaning stuff can come into the blood vessel, stuff can leave the blood vessel. Okay, here's my blood vessel. So stuff can come in and stuff can leave. The whole purpose, again, is so that we can get stuff in and out of the bloodstream. Okay? So when we get down to this capillary, it is basically just what is left of that tunica intima. We got a little bit of connective tissue that's going to wrap around, and that's the basal lamina. It's going to wrap around a tube. Okay? And if we want this tube to be the best at exchanging things, let's make it as thin as we can. So we're going to use simple squamous epithelium, and we're going to put just a little connective tissue wrapper around the outside of it. And that's going to be our capillary. We got no tunica media, we got no tunica externa, and the diameter is about that of the red blood cells size. Okay? That way we can make the red blood cells go through in single file, and we can extract as much oxygen as we can from that tissue. Okay, from that bloodstream. We've got 3 different kinds of capillaries, and I'm going to really say we have 4 different kinds, because this continuous capillary, we've got 2 different kinds of continuous capillaries. Okay, and continuous capillaries are the ones that we think about everywhere. Okay, they're found in the skin, the muscles, pretty much all over our body, and we say the endothelial cells are continuous, meaning they are all butted up to each other. Okay, they're all butted up. They're pop riveted together, like we've talked about with those desmosomes, hemidesmosomes, things like that. And we're going to allow things to get through. And I'll explain how things get through later on. Okay, but being continuous means there's no gaps, there's no pores, there's no holes, nothing. They're just complete. The surface is complete. Okay. Then we've got a fenestrated capillary. Okay, with the fenestrated capillary, the cells have pores in them, and these pores are going to mean we're going to have a lot more permeability. Okay, we're going to have large pores that are going to allow larger things to make it through than what could make it through with the continuous capillary. Okay, and then we've got sinusoidal capillaries. These don't have pores, they have gaps between their cells. Where the continuous, they all butted up, like I said. Sinusoids, they don't all butt up together. They leave gaps, which can allow the larger things to make their way in. Okay, so if we're thinking about permeability, the sinusoidal, these are going to allow the largest things through, the fenestrated, okay, the next size through, and then the continuous capillaries, they're the most restrictive. Okay, the most restrictive would be our continuous capillaries. Okay, this is really for feeding and picking up stuff and that waste. It's just your normal old capillary. Fenestrated capillaries are going to be found in the kidneys, where we need to filter. So it's going to act like a filter, a bunch of pores like a filter. Also the endocrine glands, so we can load up those larger hormones. Okay. That's going to be able to be done with pores. With these sinusoidal capillaries with their gaps, okay, it says they're found in the liver, the bone marrow, lymphoidal tissue, okay? So let's think about what y'all learned when we talked about plasma proteins, how many of the plasma proteins were made in the liver, right? About 90%. So loading up those big plasma proteins, we need big gaps to get those in. Okay, the bone marrow. What does the bone marrow make for us? That's right, we learned that in the blood. Erythrocytes, leukocytes, platelets, all those things. So we need these big gaps here. Lymphoidal tissue, this is where we're going to allow the white blood cells to crawl out, okay, and come to rest at a location. Okay, so these are really moving things that are very large. Okay, these, as I said, smaller. And then the last one, your normal capillary that's not doing big size molecular weight structures. Okay. Okay, when we look at the continuous capillaries, there's actually two types of these capillaries. Now, when we look at these two, they have the complete endothelial lining, okay, meaning if you look at the way that this is set up, all of the cells are stuck up against each other, okay? There's a good, almost like a suture line where these all come and abut one another in here. And it makes a nice, smooth surface for everything, okay? In looking at these continuous capillaries, like we said, found in all of the tissues with the exception of epithelial tissue and cartilage, being avascular this way, they're going to permit the movement of things in and out. So the way things can move in and out of these are between the cells, okay? So where this little suture line is that we're talking about, things can leak across, as well as they can move through. You know, if you're lipid soluble, you can move through. Otherwise, it's going to take some kind of a channel or something like that to allow things to move across, okay? Little things is what noticed all of these are, okay? Little things. Small solutes, water. If you're lipid soluble, like we said, and remember from AMP1, all of those respiratory gases, CO2, O2, those are lipid soluble, okay? The NH2, ammonia type things, nitrogenous amines and that kind of stuff, lipids and small alcohols, all are able to make their way through this, okay? Now, when we get to AMP2 a little bit further down the road, we're going to have to talk about a lot of these because we have a chapter set up for small solutes that are going to be things like our carbohydrates, fats, and proteins, amino acids, things like that. We've got a respiratory chapter for CO2 and O2. Okay, with the digestive system and metabolism, we're going to create some nitrogenous waste that we have to deal with. Okay, lipids also from the digestive system and small alcohols from the digestive system as well. Okay, so this is basically feeding, getting rid of the waste, okay, as well as providing oxygen and taking away the carbon dioxide. What it's going to block, it's going to block blood cells. Blood cells can't squeeze through this. Okay, the red blood cells, white blood cells, they can. And the plasma proteins. Plasma proteins are going to be too large to fit through this. Okay? So that's continuous capillary. That's the regular old capillary that we're thinking about. There is a special form of the continuous capillaries that is called specialized continuous capillaries. That's a great name for it. And we see this, we talked about this with the blood-brain barrier. We also find this in the thymus. Okay? Very restrictive permeability here. So go back and think about the blood-brain barrier. We were trying to keep things away from the brain that had a charge on it. Okay, remember you had to go through those astrocytes, that big boundary of astrocytes. So we've got these cells that are going to have much tighter junctions here, and it's going to restrict what we're going to allow across. So it's going to be even more restrictive than the continuous capillary, the regular old continuous capillary. Okay? All right, second type, fenestrated capillaries. These are the ones that have pores, and that's what you can see over here. Okay, they've got pores. All these little pores, okay, allow for us to move a bunch of larger structures through them. Okay, larger structures through them. This is going to increase the permeability of this type of capillary. So what we're trying to do is move more stuff through that has a larger size to it. Okay, it says we've got them in the endocrine organs because those hormones are going to be larger. Okay, the hypothalamus, the pituitary gland, okay, those are producing those big hormones. Kidneys, it's a filter so that things that are leaving the bloodstream, okay, we can filter, we can get rid of those waste products. Intestinal tract, that way we can move things in through this that's larger from the digestive system into the bloodstream. Okay. Then we have a type of capillaries that is called the sinusoids. This is our third type, and it's going to resemble fenestrated capillaries. We're going to find these in areas, the liver, the spleen, the bone marrow, and other endocrine organs. And what we're moving through here is much larger structures. We talked about the liver being for plasma proteins and cells and things like that making their way through. Platelets here, the spleen also, platelets, okay, white blood cells, things like this. The bone marrow, loading up red blood cells and white blood cells into the bloodstream. Endocrine organs producing those hormones, large hormones that we can move into. Think about testosterone, estrogen, things like that. They're going to get moved in the bloodstream and get carried all over the body and have different effects at different tissues. Okay, so we're not having pores here. What we have are gaps between the cells. Okay, now you would think that the sinusoids would have the greatest permeability, but that's not true. Actually, the fenestrated capillary has a greater amount of exchange, okay? Because this is a slower process. It's a slower process when it goes through. We're going to see it able to do all different sizes of things from water all the way through to plasma proteins and cells, okay? And with that, because this is a big area for things to move through, this is a place where bacteria, viruses, and things like that could make their way from the bloodstream into the body or from the body into the bloodstream. Let's put some phagocytic cells. Let's put some immune system cells here that can help us identify and control those foreign invaders. Okay, now let's look at this capillary bed. Let me describe everything for y'all right quick. We have the arteries coming in here, down, getting smaller into an arteriole. Notice we've got all the layers here, and we just got the smooth muscle band down in this area. So we know we're at the arteriole if there's just smooth muscle around it, that single layer. And notice that that continues all the way down. You can see along this edge, this is the smooth muscle. Okay, and then when we get in the capillary network, notice that we lose a lot of that smooth muscle, but we do find it in areas where there are junctions. Okay, and I'll explain that here in just a second. So there's some smooth muscle cells here and here on either side. Okay, so if we're looking at this, we call this a capillary bed. A capillary plexus is another name for it, where it's the connection between an arteriole and a venule. Okay. Now, when we're describing this, there is some anatomy to the capillary. We have this thoroughfare channel that is the main passageway through our capillary. It's a direct connection between the arteriole and the venule. And notice that there are places in here where we can do exchange that doesn't have the smooth muscle around it. Okay, so we can do capillary exchange here, but it's the direct pathway through it. Okay, so most of the blood can go this way. We're able to control these by little smooth muscle segments. That's what it's showing us with all those smooth muscles at the boundaries. So I can squeeze it down in areas. Okay, I can open it up in areas so that we can feed different amounts to different locations. Okay, We call these the meta-arterioles, these little smooth muscle segments that are going through our thoroughfare channel. Okay, notice up here at the top, we've got collaterals. We've got two arteries that are feeding this big capillary network. Okay, and that's handy because what if we get one of these that becomes blocked? I still get blood flow to the area from the other one. Okay, we call those two fusions. We call that an anastomosis. The fusion of the two vessels is called an anastomosis. So right here is our atrial anastomosis, since we have both vessels coming together. Okay, and like it says, we got good insurance if we get a blockage. Okay, there is a connection past this capillary bed. Okay, so here's the capillary bed in here, and notice I've got a larger vessel down here, and this is called the arteriovenous anastomosis. Basically what it is a way to get the blood back into the venous system if we don't need to utilize that capillary network. Okay, it's a direct connection between the artery or the arteriole and the venule. Okay, and this way, if we bypass that, if this doesn't need any more oxygen, nutrients, things like that, let's say we're at rest and we don't need to feed this at the moment, we have to have a way to get that unused blood back into the venous circulation. Notice that there are smooth muscle segments all the way around it. That's what all these little bubbles are along the outside. That's our smooth muscle bands. Okay, like you can see here. And again, we can't get exchange, so notice that we don't see the red turn into blue. Red signifying oxygenated blood, blue signifying deoxygenated blood. There are sphincters here that guard the entrance to each one. Okay, we got a little sphincter here, we've got a sphincter here. Okay, and we're able to open and close these sphincters by constricting that smooth muscle. Okay, and that's going to oppose blood flow into that area. Okay, we have a term called vasomotion for this, and that is the process of contracting and relaxing, that cycle that allows us to constrict and dilate, okay, these blood vessels. Okay, and this is going to allow us to route the blood through the capillary based on the needs of the body. Where those cells need to be oxygenated and they need nutrients and things like that, we're going to send more blood into that area. And we know this when we're at the gym and you finish working out a particular muscle group, there is a lot more blood flow in that area. We get a pump from that because we're sending more blood into the area, therefore more blood is leaking out. Okay, so you know, when I was younger, dating, things like that, you do some curls and that kind of stuff to make your biceps even bigger. And then when you arrive at the young lady's house and you go pick her up, you give her your arm and she can put her hand on that big bicep and go, ooh, look at that. Okay, it's going to go down after a while because that blood flow is going to be reduced to the area. We're going to move the fluid out. So once you feel your bicep going down, what you do is you hold her hand the rest of the night. That way she's felt that big bicep, okay? And then it loses its size over time. Then you hold hands with it, okay? At least that's what I did. Okay, so last sentence down there at the bottom, when it rests, only 25% of the vessel within the capillary bed is being utilized. About a fourth of it we're routing blood through, okay? So this allows us, this vasomotion allows us to continually adjust how much blood an area, a tissue type, a muscle, an organ even receives that much blood. How much do we send through it? Okay. And then on the other side, we've got the veins. Okay. They're going to pick up this blood that has made its way through the capillaries, or if it went through that arteriovenous anastomosis, we're going to pick it up as well. So it's going to go through the venules and then into the veins. And the role of the veins, the job the veins have is to return this blood back to the heart. Okay. Now, when we look at the arteries versus the veins, we remember that the veins are larger in diameter. Okay. They hold a greater volume of blood. That's what we need to think about. They have much thinner walls. And we've lost a lot of pressure when we make our way into these capillaries. We've had to reduce the pressure or we'll blow them out. So there's no place for us to really increase the pressure a lot in the veins. So they're going to carry a much lower pressure. And that's why that thinner wall will work. Okay, so we're on the other side. We're on the blue side now, and we're making our way back up. Remember, we've got the venules, or the smallest veins, okay, the smallest of our veins, and they've got that connective tissue wrapper around it and that kind of stuff. When we get to the medium-sized veins, we've kind of got all of our tunics back, and in the large ones, we definitely have all of our layers back, okay, and they're on their way back toward the heart. Okay, while we're talking about these veins, there are some other characteristics that we want to think about. We need to remember that veins are such a low pressure system. That whole venous side of things is such a low pressure system that we actually need valves to prevent backflow. And this shows you a couple of these valves in our picture up here. This shows you a closed valve here. Here's another valve here. Okay, so we've trapped a volume of blood between these valves. Now, one of the nice things is when we do exercise, we're going to, even though there's low pressure and there's a pressure that wants to push it toward the heart, we can increase that by doing muscle contractions. When we contract this muscle, it is going to put pressure on the veins. Can the blood go this way? No, the valve is closed, so it will only allow blood to move this direction. So, they used to tell you to avoid swelling, okay, to avoid blood clots and things like that. We need to get up and move around, and they would tell people to get up and move around the airplane cabin, okay, during a flight. And now they want to really restrict us on that after things that have happened security-wise and things like that. When we look at it under a microscope, you can see this darkening. This is a valve here, and this is a valve here in our slide of a capillary, okay? Now, occasionally, over time, we can wear out these valves and they can become leaky valves, okay? Which means we're not going to be able to push this blood the way it normally would. Blood's going to pool down here, and that's what we call varicose veins, okay? It's the same thing that we see with hemorrhoids, just in a different location, okay? I love this graph, this pie chart here. It shows us the blood distribution based on arterial versus venous blood. And if I ask you, if I just said, okay, is there more venous blood? Is there more blood in the venous system or in the arterial system? Okay, we want to think, okay, we want to be feeding more of that. Okay, so there should be more in the arterial, but that's not the way it is at all. Okay, from about here, half of the capillaries in the lungs should be deoxygenated blood, okay? And then the systemic capillary, half of that should be deoxygenated blood. So look at all of this area, all the way around to here, shows us deoxygenated blood, okay? That venous system, okay? Just from here to here, 64%, okay? And then if you add this portion in where the capillaries and the arteries are that are carrying it, we're probably at 2/3 of it is deoxygenated blood. Well, how does that work now? Okay, well, remember, it's under much lower pressure, and those veins have a greater volume to them. Okay, they have a bigger diameter, so we're going to find more blood in the venous system than we do in the arterial system. Okay, that brings us to cardiovascular physiology. This is the physiology of our circulatory system, okay? It's how are we going to maintain blood flow? It is how do we direct the blood flow through the body, okay? And we've got a couple of things that we need to think about. We need to think about pressure. That's going to be generated by the heart. And then we've got resistance, okay? Resistance is things that are going to reduce the pressure. It's things that we have to overcome with pressure. Okay, now look at our graph over here. Remember that the heart's going to play a role in this as far as generating the pressure, and then we have the ability to maintain that pressure based on smooth muscle in the arteries. So we have a control mechanism here, basically okay, with squeezing it down. We also have to deal with our brain and our endocrine system as far as they're having a control over our circulatory system. Okay, now all of this has to be maintained so that we can, at the capillary, do exchange correctly. Okay, because if the pressure is too high, we'll blow stuff out. If the pressure is too low, we won't be able to send enough across. So that becomes an issue. Now, lesser issues we have over here on the venous side because we don't have the control over the venous side. Remember, that smooth muscle band is very small in the venous side. Okay, so we don't have the ability to control it like we do the arterial side. So most of it is going to be over here on this side of our paper, not as much over here on this side. But it's going to play a role. Don't get me wrong, it will play a role. We've been talking about this pressure, okay? So let's think about this pressure for a little bit. Go to the doctor's office, they take our blood pressure. Go to the dentist's office, they take my blood pressure at the dentist now, okay? What we're looking for is, are we out of homeostasis with respect to our blood pressure? But that's only one of the types of pressure that we really look at. In this whole circuit, there's going to be differences in this pressure. What's going to happen in your arm is going to be different from what's down at the capillary bed, okay? So we've got blood pressure. That is our arterial pressure. We do that usually in the arm. Pump it up, occlude the blood flow through it, and then wait till the heart can beat that pressure that has been generated by squeezing it with the cuff. Okay, we're measuring that pressure in the cuff so we can figure out what the pressure is that's beating it. Okay, we hear it real loudly when it is being taken, and then when we don't hear it anymore, we know we're no longer under pressure. So what we want to hear with this is blood flow, want to hear our pulse, and then we don't want to hear the pulses loudly. Okay, so that's arterial pressure. Okay, capillary hydrostatic pressure, that's the pressure that's inside the capillaries, okay, of your body. Now again, if it's too high, we can blow these out, okay? So we don't want that pressure to be as great as we would see in the arterial pressure, okay? And then the venous pressure, that's going to be very low because we've been through the capillaries, we've reduced that pressure down so much that now we don't have a way to increase the pressure. In fact, we're going to continue to lose pressure because of resistance. Okay, and we'll talk about resistance coming up. So this pressure is going to change over the course of this circuit, this systemic circuit that we talked about. It's going to change. In fact, we're going to lose about 100 millimeters of mercury worth of pressure. Okay, now that value relates to the pressure that we talk about when we say blood pressure 120/80. Okay, so we're going to lose about 100 millimeters of mercury. So if it's normally 120, then we're going to lose down to 20. Okay, we're going to have a very low amount of pressure by the time we're done. Okay, so this pressure is being reduced by something called peripheral resistance. Okay, peripheral resistance. And we say that there is a resistance in this entire cardiovascular system. Okay, so let's think about how this resistance works. Resistance, or vascular resistance, we use as the letter R. Okay? Now, when we think about blood vessels, the blood is going through the blood vessels and it's going to keep bouncing into the walls of the blood vessel. That's going to slow things down. Imagine if you're walking through a hallway and there's a whole bunch of people in the hallway. There's a lot more resistance that we have to deal with. Okay, so the same thing here. So this resistance is going to depend on three things. One, how long is the vessel? The longer the vessel, the more we're going to bump into the walls. Number two, the diameter of the vessel, how big or how small it is, that's going to determine how often we run into the walls. And lastly, okay, that pressure that we start with, okay? So the pressure is going to be dependent on the length of the vessel as well as the diameter of the vessel. Well, by the time we're an adult, like we are now, our vessel length is constant, okay? We're not going to grow anymore, so our vessel length is constant. So we can kind of eliminate what's going on with vessel length now. So what do we have left? Vessel diameter. And we know that we can control the vessel diameter, our arteries, okay, especially because we can do vasomotion, that vasodilation and vasoconstriction of things. Now, something to keep in mind is when we start constricting a vessel, okay, that is going to increase our resistance. We'll say that again. As we constrict the blood vessel, it is going to increase our resistance, meaning, okay, and we know this, if I put the end of my thumb over the water hose and I constrict the flow, it's harder for that water to get out, okay? It takes more pressure. Pressure has to build up behind it in order for it to make its way through, okay? So this R is going to increase exponentially as vessel diameter decreases. So as we squeeze down, we're going to see a doubling. Okay, that's what exponentially means, a doubling of the resistance. So for each unit we squeeze, okay, we're going to increase the resistance twice that amount. So we're able to make minor adjustments that have major effects over resistance, okay? This shows you the pressure in the systemic circuit. This is another graph I think that is a really good graph for us, okay? Really good picture. So let me explain. Here we've got, remember the pressure 120 over 80, okay? So this is systolic pressure when the heart is squeezing, diastolic pressure when we are in diastole, okay? So we see changes, and we know that. We see squish out, relax, squish out, relax, right? As it's leaving, as it's in the blood vessel, okay? Well, we can kind of, oops, I meant to erase that. We can kind of standardize the line. We can make it more linear if we do something with pulse pressure, okay? Where we just take the difference between the systolic pressure and the diastolic pressure. So let's even it out, and we're going to make this our line. Okay, here's our line, and that way we don't have to mess with this and this. It's kind of an average, okay? That mean arterial pressure whoosh, goes like this, okay? So if we do that, we said we're going to lose about 100 millimeters of mercury worth of pressure. Well, for us, we have to be careful because look, we're starting out at around 100, but by the time we make our way through the system, we are down to, that's right, 0. So we've lost almost 100 millimeters of mercury worth of pressure, okay, as we've done this, okay? So it says we're in the aorta here. Remember, the aorta is very stretchy. It's got a lot of elastic fibers in it. So as we hit it and we hit these elastic fibers, we actually see an increase in the systolic pressure. Well, why does that happen? Well, it happens because those elastic fibers are pushing against it, and that's going to increase the pressure, okay? It's like squeezing on a balloon. If you squeeze on the balloon, it's liable to pop, right? So the same thing here. These elastic fibers are pushing against it like our hands are pushing against it, okay? So we actually see an elevation of the pressure inside the elastic arteries. But by the time we're out here in the muscular arteries, we're back down to where we need to be. And that's why we take our blood pressure in the arm and in the leg if we don't have any arms, okay? So that works. We get a good record of what the heart is doing as it leaves if we're measuring it where the muscular arteries are. Y'all see that, right? And then as we keep branching and branching and branching and branching, volume increases, pressure goes down, okay? Meet more resistant, pressure goes down until we make our way into the capillaries. By the time we make our way into the capillaries, we need to be down to about, let's say 35, okay? 35 millimeters of mercury entering the capillary. When we leave the capillary, we're gonna be under 20, okay? Maybe 18 or so. And then there's no way to build it up. So look what happens to pressure. Venules, it continues down, medium-sized veins, large veins, vena cava, and then bam, down into make its way back into the heart. When it makes its way back into the heart, we're almost at a pressure of 0, okay? Almost a pressure of 0. All that this is mean arterial pressure. This is a way to create that line that we were talking about to kind of make it a linear event. And I'm not going to ask you any questions about mean arterial pressure, okay? So if we have this normal pressure, 120 over 80, if we're above that or below that, we're outside of homeostasis with respect to blood pressure. If you're over 140, over 90 millimeters of mercury, systolic versus diastolic, that's considered hypertension or high blood pressure, okay? And it's causing your heart to have to work harder. We're generating much greater pressures. That means we're squeezing the heart a lot more, which means we're wearing the heart muscle out, okay? And then hypotension is an abnormally low pressure. That abnormally low pressure, and notice there's not a value for it because it would really be dependent on the individual, okay? And lots of things go into these values. My wife's blood pressure is much lower than mine, okay? She's a lot smaller than I am, okay? And in fact, what her normal blood pressure is, it might be hypotension for me. Okay, so we were talking earlier, those arterial walls are gonna be nice. They're gonna help push against the blood and keep the blood flowing, okay? So they stretch during systole and they rebound during diastole. They absorb that big wad of blood that makes their way in from the heart, and then they squeeze on it, okay? And basically what this does is keep that blood flowing even during diastole. We continue to force that blood along because of the elastic rebound that we have. Okay, so when we're looking at this pressure, okay, the pressure in the system, we looked at that graph, it said that it's going to decrease with the distance we move away from the heart. Okay, pressure gets lower as you move away from the heart because we're running into more and more resistance. The more we move away from the heart, the more resistance there is. Okay, so this blood pressure decreases as it overcomes the friction that we see, that resistance. Okay. By the time we're in the venous side with the venous return, that's how much blood is making its way back into the right atrium each minute. So we have a calculation for that, okay? And again, that's going to be determined by all the way over on the arterial side, the heart's rate, as well as what's going on with blood vessel dilation and constriction and things like that, okay? And finally, the venous pressure. What are we at when we get done with the capillaries, okay? Remember that venous side has a very low effective pressure. It is very low. And we don't have really a tremendous way to increase that. Now we got some tiny things that we can do, and I'm gonna explain those to you, okay? That amount of blood that makes its way back to the heart each minute, that venous return, is assisted by a couple of things, okay? We call one the respiratory pump. I wish I'd have put that in yellow, okay? The respiratory pump. Simple breathing helps us move the blood along. And then if we get up and we move around, we talked about compression of skeletal muscles. Remember that the venous side has those one-way valves, and it's going to squeeze on the vessel if we're doing muscle contractions. That's what those wraps that they put on our legs and pump them up, okay? If you're in the hospital, that's somebody that's laying in the hospital. We want to continue this circulation, not allow blood to pool and ultimately clot. So we try to get our patients up very quickly after we have had an operation. And if they're not able to get up, or if they're bedridden for a while, we're going to maintain those leg compression, okay? So let's look at both of these. So besides that squeezing of the muscle, skeletal muscle, when we're up moving around and that kind of stuff, we also can take advantage of the respiratory pump, okay? And that's just simple breathing, because when we breathe, we see the thoracic cavity increase, it expands, and then when we exhale, we see the contraction of that thoracic cavity, okay? So when air comes into the lungs, it actually increases that cavity, which allows us to pull blood into the inferior vena cava and the right atrium. And then as we compress that, as we exhale, it's going to decrease the size of that, which is going to help us to push the blood into the right atrium. Okay, so we kind of pull it up as we inhale, and then we kind of push as we exhale. Okay, so just breathing allows us to help with this. Okay, so the respiratory pump plus the skeletal muscle movement and things like that really help force blood along, and that's why they're going to get us up. That's why we do breathing treatments. That's why they get us up in the hospital and things like that. Make sure we got good blood flow. Okay, let's talk about capillary exchange now. Okay, we got all the parts, we got the pieces down. Let's talk about the most important part of this chapter, how the capillaries are able to exchange stuff from the inside to the outside. That would be in the blood to the interstitial tissue or extracellular fluid. And how the extracellular fluid or interstitial tissue, or interstitial fluid, excuse me, can put stuff back in the bloodstream. So it's going to be a two-way street. So this capillary exchange is vital. That's the whole reason why we have a circulatory system. We can move stuff around, but we have to be able to get it out of the circulatory system. We have to be able to get new stuff in and get the other stuff out, okay? Nutrients got to come out, oxygen's got to come out, hormones got to come out, but then we also have to be able to put these hormones in, we have to be able to put these nutrients in, okay? We got to be able to take the waste out, we got to take carbon dioxide into the system, okay? So lots of things going on here. We say that there's three main physiological activities going on here, okay? First one's diffusion. We learned about diffusion way back in A&P 1. Diffusion is going from a high concentration, low concentration, okay? That's the way nature does it. Nature tries to even things out. So if there's more on one side than there is on the other, and it can go through, if there's no barrier to this diffusion, then things can diffuse, right? So we talked about lipid solubility in A&P 1. We talked about the plasma membrane as being selectively permeable, right? So there's certain things that are going to allow for diffusion to work, okay? And we're not going to have to worry about pumping it across or anything like that. The second one is filtration. The filtration is, there is a filter with filtration, okay? There is this porous membrane that things have to be pushed through, okay? If you're too big, the filter stops you, right? And then reabsorption. Reabsorption says we're going to go the opposite direction of filtration, okay? So let's take a look at this. Start with diffusion. Y'all know, movement of ions, molecules, stuff, I like to call it, from a high concentration to a lower concentration. We say it moves down the concentration gradient, okay? We got 5 routes that things can diffuse through. 1, water, ions, small solutes, glucose, things like this can go through those little fenestrated capillaries. Remember, there's pores in them. These guys are small enough they can go through the pores. It's not going to be a, it's not going to prevent diffusion from happening. So if there's more stuff on one side than there is on the other, because they're small enough, they're going to be able to go through. Also, in those continuous capillaries, okay, there is adjacent cells where they are put together. So there's little tiny gaps between the cells. Things are going to, the small things are going to be able to squeeze through where these cells don't overlap one another. They just butt up against each other, okay? So that's route number 1. Route number 2 says we've got ions like sodium, potassium, calcium, chloride. We've talked about all of these in A&P 1 that have channels that can go through. Remember, there are channels in the cell, proteins, okay, membrane-bound proteins that allow these things to go through. So that's another way to go from high concentration to low concentration. There's a channel, okay, built by a protein. It's not a pore, it's an actual channel, okay? And then we've got large water-soluble compounds can go through those fenestrated capillaries, okay? And I'd go ahead and include sinusoids here as well, okay? There are large pores or gaps, and these guys that are large, as long as you're small enough to fit through the hole, you can go this way. So it's going to go from high concentration, low concentration again. Number 4, lipids and lipid-soluble things. Remember that plasma membrane is not It's not a barrier to this, okay? So oxygen, carbon dioxide, alcohols, things like this are lipid soluble. They will be able to go straight through, okay? And the last one, those plasma proteins can go through the sinusoids as well. These are big, okay? Cells, plasma proteins, again, can diffuse into the bloodstream or out of the bloodstream depending on the size of this hole, okay? So plasma proteins are things that we're going to want to move in as well as move out, okay? So these are our 5 diffusion routes. And what we're saying with this, kind of the big take-home message is there's 5 ways that we can move stuff across without involving anything else. It's just going to naturally occur. If there's more oxygen on one side than there is on the other, oxygen is going to go from high concentration to low concentration, right? Carbon dioxide is going to go from high concentration to low concentration. If you drink a bunch of alcohol, alcohol is going to leave the bloodstream and it's going to affect your brain, okay? Big plasma proteins. They don't fit through any of these small guys. It's got to be at a sinusoid for us to get plasma proteins. It's got to be cellular almost, okay? Big cells like in the bone marrow to get the cells into the bloodstream, okay? So that's diffusion. So we got diffusion down. Let's look at filtration now. Filtration, one important thing with it, pressure. It's going to involve pressure. And the pressure that we are talking about is the pressure exerted from the heart. And we make our way down into the capillary, so there is this pressure that's left over from the heart pushing, and that's called capillary hydrostatic pressure. So there's a greater pressure on the inside of our capillary. Here's our capillary. I'm going to draw a tube for us, okay? Here's our capillary, and notice that we've got a continuous capillary. They're showing us the regular old kind. Here's our cells, and there's little spaces between that we're going to allow things to be able to fit through. If you're small enough, okay, you can fit through that hole, and there's a pressure that says I'm going to force things through, okay? In my house, I got to clean the filter for the air conditioner, right? And the way that that works is the thermostat kicks on, and it starts a pump, okay? And that pump creates a pressure that forces air through. It pumps air out to the air conditioner to be cooled, right? And then back into the house. And by doing this, it creates a suction through where this filter, these things must be filtered. And I got dogs, and the dog hair fills the filter because the hair is too large to fit through the porous filter. So there's a pressure involved here, okay? The difference between filtration and diffusion would be like me just putting that filter up and there being no pressure. I'm not going to gather a lot of this hair, right? So notice down here in this, if you're small enough, you fit through. If you're too big, you're not going to fit through, okay? If you're this big around, you don't fit through that hole. Doesn't care what the pressure is, it's not going to put you through there, okay? So that's what we consider filtration. It's under a pressure. And the pressure that we're talking about originated up at the heart where we had a pressure of 120 over 80, right? By the time we get down to the capillaries, remember it was down at what, about 35 we said when it made its way into the capillary, okay? So there's still pressure there that's going to force it across. Okay, so filtration is the removal of large solids through a porous membrane driven by hydrostatic pressure, okay? At the capillary level, lots of things can be filtered. Water, small solids, it's being forced. It's not diffusing, it's being forced because there is a pressure, okay? Now, if you're too big, you get stuck in the bloodstream. You don't get to be filtered out, okay? Okay, reabsorption. Okay, reabsorption is very similar to filtration. It's going to deal with the pressure, but this pressure is due to osmosis. Okay, we call it osmotic pressure. Okay, and it's due to there being stuff in the water. Now, in A&P 1, I had a tube like this. Okay, and we talked about, here's my tube. It's a U-shaped tube, and first of all, I'm going to put a solid piece of glass between these two. And in this side, I'm going to pour a 70% glucose solution. And this side over here, I'm going to pour a 30% glucose solution. Okay, more glucose over here, less glucose over here. Where's there more water then? More glucose here, less water. Less glucose here, more water, right? So this part has more water in it. Okay, more H2O. So we're going to pull right here. Fill this side up 30%. Fill this side up, made it equal amounts. Just like that, 70% glucose over here. Okay. Now, I'm going to withdraw my plate here in the middle. Okay. And when I do that, there is a screen between them. Okay. When I remove the plate, there's still a screen there. And that screen has little pores in it. Okay. Didn't really want to do that, y'all. Hang on, let me fix it. Okay, so now you can see we've got a porous, almost like a sieve here, right? A filter here, okay? So now what's gonna happen, okay? Now what's gonna happen with our water? Well, water's small enough to fit through here, and anything that's small enough, but the glucose molecules, they're way too big. They're like this big, y'all. So they can't go across, okay? And remember, there's a whole lot more glucose molecules over here because there's 70% of them over here, right? So we got tons of them over here. More water over here, less water over here. So which way does water go? Water goes from high concentration, low concentration. It's able to go through this, and that means this side of the tube fills up more. We lose part of it from this side. Let me try to do it this way, y'all. Oh, I feel like I'm pretty smart doing that. All right, we're going to lose some here. We got more over here. We've got less over here. And now what's the concentration? Concentration on this side, 50% glucose and water. Side over here, 50% glucose and water. Okay, now, what if I put a plunger over here on this side? Put a plunger here. Could I push down on this and force water back across? Okay. Yes. Okay. I could do that. Okay. So that pressure that it takes to stop this, okay, going back across, means that there's a pressure that happens when we moved it across. We call that osmotic pressure. Okay. Pressure that is required to prevent osmosis from happening, osmotic pressure, okay? So the way we're going to think about it is in our tube, there were more glucose molecules over here. There were less over here. There was more water over here, okay? So what caused the water to go from high concentration, low concentration? Osmotic pressure. These guys, there's more of these guys in here, there's less water. It says, give me some water. Okay? So that's osmotic pressure. And we're going to use that. That's reabsorption. Okay? So y'all agree that there's stuff in the blood. There's stuff in the blood that's too large to go across the capillaries. Are there plasma proteins? Are there cells? Are there platelets? Are these big things that are there? Yeah. And there's not those big things on the other side, right? We don't have plasma proteins on the other side as much. There's a lot more in the blood, okay? So the blood has this stuff. The stuff is called the colloid that's in there. So we call it the blood colloid osmotic pressure, the BCOP. That means there's a lot more stuff in the blood than there is out in the interstitial fluid. Okay? Hydrostatic pressure, we talked about with filtration, it forced water out. Osmotic pressure, because there's all of that stuff inside the blood and there's not outside the bloodstream, it tends to pull things in. Filtration pushes the water out. Osmotic pressure pulls it back in, okay? And that's what we're dealing with capillaries, okay? So we got to be able to talk about the pressure due from the heart beating, that's our hydrostatic pressure. And then the osmotic pressure, the pressure of the stuff that is inside the blood pulling the water and the other stuff that's small enough to fit through back in, okay? So here's what it looks like, okay? Here's our old buddy, the capillary. Here's our arteriole over here. Here's our venule over here. And in between that, this is where the actual capillary is, okay? Now, when we look at the difference between the inside and the outside, that's what we have to look at here. When we get to this point right here, in here, there is a pressure that is pushing out that comes from the heart that is 35 millimeters of mercury, okay? And that should be capillary hydrostatic pressure. This key is wrong. Don't look at that, okay? This says there is pressure pushing it out, okay? Excuse me. Okay. And there is also stuff in the blood. There's more stuff in the blood than there is out here. So there is a pressure to want to draw it back in. That's called the blood colloid osmotic pressure, right? Well, look at this. The pressure pushing it out is 35. The pressure pulling it in is 25. We get the winner being capillary hydrostatic pressure. Capillary hydrostatic pressure is greater than blood colloid osmotic pressure. So the force pushing out is greater than the force pulling in. So which way does the net flow go? It goes out. It goes out, okay? And as this fluid leaks across, okay, fluid leaks across, remember, pressure is going to decrease. So it's going to, as we move along and we're losing volume, pressure goes down, pressure goes down, pressure goes down, pressure goes down. And we get to this point here in the middle where notice the pressure pulling it in, 25, the pressure pushing it out, 25. It says no net movement occurs. Now that's an interesting term, no net movement, which means we can have movement, but if you have as much moves out, the same amount's going to move in. So no one wins is what we're saying, okay? And we call that the transition point. This is the transition point right here of our capillary. Now, remember, as we continue to go this way, we keep running into more resistance, okay? So the pressure continues to drop on the inside, and it keeps going down, and it keeps going down. And notice we get to this side over here, and now our capillary hydrostatic pressure is 18. Our blood colloid osmotic pressure, there's still the same number of big things inside the blood, so it's still 25. So now, who's winning? That's right. Blood colloid osmotic pressure is winning, so we're going to move things into the bloodstream over here. Now that's pretty cool when you think about it, okay? What we're saying is, in a capillary that is functioning in homeostasis, okay, this side of it is going to push stuff out. This side of it is going to pull things in. And here in the middle, we run into that transition point. And I said middle, and I really shouldn't have said middle, okay? Ignore me saying middle. I did not say, well, I said middle, okay? But I shouldn't have. Okay, because let's look at this, okay? They drew this line here. Is this line longer than this line? And it is, okay? It is. The amount of filtration that happens is always greater than the amount of reabsorption. So this really is a little bit toward the venule side, okay? Transition point is a little bit toward the venule side. And that's going to be important, okay? That's going to be important, and I'll explain why, okay? But this is important, okay? This is important. We're getting to exchange here, okay? the stuff that's in the bloodstream is getting filtered out, okay? That means if we've got nutrients, if we've got ions, if we've got small hormones, if we've got amino acids, whatever it is that we've got, oxygen, whatever it is that we've got here, we're able to push it out. And then once you get toward the end over here, we want to draw things back in, okay? Now, what are we drawing back in? Well, there's more waste out here. There's carbon dioxide out here, okay? There's fluid out here. So all of this is going to get to move back in, okay? But we're not going to do it perfectly. We're going to push more out than we pull in, okay? So so how does that work, okay? Why are we not just swelling up all over the place then, right? Because if things are leaving the bloodstream and moving into our tissues, why? And that's what swelling is. That's edema, extra fluid in your extracellular fluid or interstitial fluid. And it came from the blood. Okay? Why? What do we do with that extra fluid? Okay? What do we do with that extra fluid? Well, we have a whole system designed to do that. Here's the deal. Last time I flew over to Scotland, we went to Scotland for New Year's this year and got on the plane. It's a long flight, and I'm a big guy, you know, 280 pounds, twisted steel and sex appeal on the plane in this, and we can't afford it. I work at a community college. We can't afford to fly first class, get a big seat. So I'm in one of these little seats. My wife did give me an aisle seat where I got a little bit more room, but I'm still a big guy, 275 pounds, twisted steel, sex appeal, pushing down on me in the seat, right? Okay. And they make us sit in that seat. Don't get up, move around the cabin. Get back to your seat, right? So I notice my feet start swelling, okay? I can feel it. My shoes get tighter on this plane ride. After a few hours, I have to loosen my shoes up a little bit. And I knew at that point what I needed to do. I needed to get up and stretch my legs, because what I had done is I had compressed, not my blood vessels, I had compressed my lymphatic system. My lymphatic system wasn't draining the same way it should, because all of this extra fluid that makes its way into the interstitial fluid ultimately ends up in the lymphatic system. And what we do with it is we send it back into the bloodstream, okay? So the extracellular fluid ultimately makes its way back into the blood via the lymphatic system, and it travels through a bunch of these lymph nodes where we can check to make sure there's nothing in us that shouldn't be in us. We can check for foreign bodies. That's what the lymph nodes do. They clean our lymph, okay? So let's talk about this, okay? The hydrostatic pressure, we're looking for what's going on between capillary hydrostatic pressure. That's the pressure that the heart has created, pushing stuff out, and the colloid osmotic pressure, that's pulling stuff in. Now, this talks about there being interstitial colloid osmotic pressure and an interstitial capillary hydrostatic pressure. Those are negligible, y'all. The ones that we need to deal with is capillary hydrostatic pressure and blood colloid osmotic pressure. And all we got to do is know which one's greater. If capillary hydrostatic pressure is greater than blood colloid osmotic pressure, we're doing filtration, we're pushing stuff out. If blood colloid osmotic pressure is greater than capillary hydrostatic pressure, we're pulling stuff in, okay? So what I was saying is the direction that things are going, where the pressure is winning, is really, we can disregard this one, we can disregard this one. We got capillary hydrostatic pressure versus blood colloid osmotic pressure, okay? If this one's winning, if this one's larger, we're pulling stuff in. If this one's larger, we're pushing stuff out, okay? So we get movement out of the bloodstream into the interstitial fluid. We get movement out of the interstitial fluid back into the capillary where the blood is, okay? Arterial end is where we do filtration. Venual end, that's where we're doing reabsorption. That point that's not quite in the middle, that's a little bit closer to the venule side, that's called our transition point, okay? And that's, so if we think about it, if this is the length of our capillary and our transition point is here, notice we're going to filter more than we're going to pull back in. We got to do something with this excess, okay? And it says the excess ultimately enters the lymphatic vessels and it travels around the body, okay, in the lymph, and then it dumps back into the bloodstream. And the cool thing is, next chapter we're going to cover is the lymphatic system, so we'll see exactly how this works. What this does, what this excess of it moving out does, is it constantly sends stuff to the interstitial tissue. It constantly feeds our tissue more than we collect the waste, okay? So we have people that their kidneys aren't functioning and they have to go on dialysis, okay? Well, dialysis is one of those things where you may have to go every other day, every third day, depending upon functioning of your kidneys and that kind of stuff, because we can handle having waste in our bloodstream, okay? We got to feed our tissues though, right? So it's much more important for us to feed the interstitial fluid than take back really the waste that we're talking about, okay? We can do a little bit better with it. So it says that water is continuously moved out of the capillaries, okay? And then the lymphatic system brings it back and puts it back in the bloodstream. So what this does, it ensures constant plasma interstitial fluid communication, okay? It means that the blood is always talking to the interstitial fluid. We're getting good turnover of this, right? Fluid is making its way from the bloodstream into the interstitial fluid, and then ultimately into the lymphatic system, back into the bloodstream, okay? Over and over and over again. Because of this, because there's more filtration than reabsorption, we get to accelerate the distribution of nutrients, hormones, and dissolved gases through the tissues. We do better at getting stuff out than we do pulling it back in, okay? Number 3 says we can transport insoluble lipids and tissue proteins that can't cross the capillary walls, okay? We have the ability to move things in our lymphatic system, okay? We don't have to put it in the bloodstream to move it around. And the last one says, because I've got this excess, I'm able to clean them with the lymph nodes. And that's where we're going to get rid of the toxins. And we're going to have tons of lymphocytes and macrophages, monocytes in our lymph nodes that are checking things over, okay? Checking that lymph over to make sure we don't have any problems, okay? There's no foreign invaders there. And if there are, we turn on the immune system. Okay, so let's think about some things. What if, let's see, we were in a car wreck. Okay, I was in a car wreck. You don't have to be in a car wreck. I was in a car wreck, and I hit the steering wheel. I didn't have my seatbelt on. I banged around inside the cab and that kind of stuff a little bit. And I had a bunch of very small breaks in my circulatory system, okay? So I'm leaking blood into my body is what I'm getting at, okay? What's that gonna do at the capillary? Okay, well, think about this. If I'm losing blood, the volume of my blood's going down. What does that do to pressure? That's right, pressure goes down too. So as my pressure goes down, what's gonna happen to that capillary hydrostatic pressure? That's right, it's gonna go down. What's that gonna do to the transition point if capillary hydrostatic pressure goes down? Okay, what we're saying, here's where the old transition point is. If capillary hydrostatic pressure gets less, I'm not pushing as much across. That moves my transition point this way. So now the transition point is here. And what do? That's right, I pull in more fluid. I pour in more fluid from my interstitial cells, more extracellular fluid. And what's that gonna do? It's gonna try to keep my pressure up. Okay, we call this the recalling of fluids. Okay, and this is coming from what would ultimately become lymph later on. We're just putting it back into the bloodstream at the level of the capillary because that transition point got moved this direction. Okay, let's say we're dehydrated. Okay, we're dehydrated. All right, here we go. Now we're dehydrated. Here's my transition point under normal circumstances. Okay, now there's more stuff inside the blood. There's less water, more stuff. We'll put more stuff, that means there's less water. What's that going to do? Okay, again, it's going to cause the transition point to have to move this way because I'm going to recall fluids again. Okay, I'm going to accelerate reabsorption because I have an increased blood colloid osmotic pressure. Okay, my BCOP is greater. It's not 18 like it was. Okay, let's say it's 25. Well, that means we're gonna move the transition point a long way over here, and that's going to allow me, if I'm dehydrated, to try to keep pumping my blood. This happens in Texas all the time. We get people with heat stroke and they die because they're dehydrated. Okay, the reason, and what happens is we keep sweating and we keep sweating. Well, where does the sweat come from? Well, the sweat comes from the interstitial fluid. Well, where does the interstitial fluid come from? Well, the interstitial fluid comes from the plasma. So all of this is linked together. So when you're dehydrated, is your blood thick? Yes. Does it have as much fluid? Does it have much water in it? No. Okay. Is it going to be harder to pump thick stuff through your blood vessels? Yes. Is your heart going to have to work harder? Yes. Okay. So with it working harder, are we creating a really bad situation now? Okay. We can't cool the body enough now because it's too thick. We're too dehydrated. Okay. And finally, the body says, I can't lose any more water. We stop sweating. When we stop sweating and we're really that hot, that's when we're in a critical time because what's going to happen to our body temperature now? And there have been people that they found after football practice, kids that went home, body temperature is 110, 115 degrees, laying in the bed. Okay? Don't do that. Drink water. The third one, okay? This is what I did on the plane, okay? I had no way to move stuff into the lymphatic system, okay? So with this, my capillary hydrostatic pressure was greater, okay? Normally it was here. We move that transition point this way, okay? That means what am I doing here? I'm filtering a lot more stuff out. I'm filtering more than I'm able to pull in. That sets the stage for edema, okay? That sets the stage for edema. Okay. This is the last information for our first lecture exam, okay? Whole cardiovascular system is going to be involved. Blood, heart, blood vessels, okay? Make sure that you've got how these capillaries work down. If you have questions, let me know.